Thank you very much. So today, mainly I'm just going to present you a research protocol that we're about to start hopefully in the next month for pending IRB approval. And I would really like to hear your thoughts and comments on that. I have no disclosures related to this subject. And before starting, just like a little bit of background, right, why we decided to do that. So as all of you know, right, every primary brain injury will be, most likely will be followed by secondary brain injury. And this happening why, right? Because like there is a huge cascade of events after the primary brain injury, depending on the severity of the primary brain injury. It can be glutamate mediated, oxidative mediated, multiple type of, different type of strokes, right? Changes in metabolism. And the question is, why is this important? It is very important because it has been shown to be related with worse outcome and increased mortality. So our goal when we take care of these patients, first is how to detect secondary brain injury, and of course, if we can prevent before this happening early on. Classically, we do that by serial neurological exam, by a combination of multimodal monitoring, invasive and non-invasive. However, up to now, little is known and little is understood about the mechanism of recovery and of brain recovery, if I can say like that, independently of the initial injury. Still, we don't know even subarachnoid TBIs or traumatic or non-traumatic, sorry. We don't only know the mechanism of recovery, why some patients recover and why not, despite having the same treatment. So, that say, it's very also important to understand first the mechanism, and that will make us understand if we can reverse the brain injury that's gonna lead to a recovery. And to this point, there was a very big, very intriguing study published in 2019 in Nature in animal models that showed that brain injury, so thought permanent brain injury, might be reversible. That was like a very intriguing study, very interesting that they did in 2019. Some of you may be familiar. They have taken post-mortem pigs' brains, and then they created the Beck's perfusate, so-called, that they had different kind of energetic substrates, and they fuse these brains post-mortem up to 6 hours post-mortem. And then what it's been shown interestingly is that the brains that they infused with this perfusate, they showed circulation, so they did have flows in Doppler with good systolic and diastolic flow in all the vessels territory. That was like a very good circulation, like there was like a functional circulation. They did different maneuvers like compression, pre-compression. You can see there's like a cortical vein filling, and also giving imodipine, see that there was like a vasodilation and both increasing flows. And to go further, what they even did more, they showed that brains that they infused with this perfusate, they had reversal of apoptosis. So here they presented in their paper both the cortical area, the C1 layer, and the dentate gyrus. And here is 1 hour postmortem, 10 hours postmortem, control perfusate, and the magic perfusate, the Beck's perfusate. And they show here in, when they stand for CASP3 for apoptosis, here if you can see the apoptosis is much less, whereas here, like in 1 hour postmortem, we see that of course there is apoptosis in both areas, but here is like there is almost no apoptosis. So they started creating hopes that maybe this so-thought secondary brain injury is not that permanent if we find the good way to reverse it. Of course, it is not that simple, right? It's extremely complicated. It's been like decades of research, and nothing has been really very small part has been understood so far. And I think like things are even more complicated if we take into account that neurons do not respond to stress the same way, and even cells in the neurons do not respond to the stress the same way. So we know, like that's been like decades known that neurons are like very selective, show very selective vulnerability to stress. And you can see like we all know hippocampus, for example, is much more vulnerable to stress than the cortex, and even in the same area of the brain, like in the hippocampus, not all the layers are the same vulnerable to stress. And to make things even more complicated, that was a table did when I was doing my research, when I was interested to see what type of stress does what to what type of neurons. We see that every type of stress do not affect the same way. So hypoxia does not affect the same neurons or the same order of neurons than, for example, CO intoxication or hypoglycemia. So that's also maybe the same thing, like in subarachnoid ICH stroke, depends on what is the primary stress that they cause to neurons. They may not affect the same way at the end the cells and the neurons, and that's why the recovery is not the same. So to that problem comes the biomarker. So clinicians try to, by doing translational research, try to understand what's going on and try to link this animal data or this data with human, right? And then the last decades, they're discovery of a lot of biomarkers, thousands of biomarkers that they reflect even astrocytes, neurons, inflammation. There can be metabolism, proteins. So they are reflective of multiple different functions in the brain. However, despite this discovery of all these biomarkers, so far nothing has been standardized. And even a couple of years ago, Neurocritical Care Society recommended against the use of biomarkers for prognostication, other than the neuron-specific enolase, which is the only one that we use for prognostication in cardiac arrest, and yet not alone. It's in a combination with other evidence. So more recent, despite Neurocritical Care Society do not recommend against, still people do not lose hope on biomarkers, and a lot of nice research on that. More recent was last year, a very nice paper from MGH that published in subarachnoid population that they found these three metabolites, the SDMA, the MGV, and ornithine, that they are all three involved in nitric oxide metabolism. Increased level of these metabolites, they're associated with poor outcome in subarachnoid hemorrhage. And to go further, we know already a couple of years ago that indeed metabolism, there's metabolic changes that may be responsible for poor recovery. That was a very nice study published 10 years ago in Brain, showed that the switch to anaerobic metabolism, that is reflected by increasing lactate to pyruvate ratio, is associated with poor outcomes. So they had 223 patients on microdialysis, and they saw that the patients with increased glutamate and increased LPR, they had higher rate of mortality, showing that indeed there is a switch to metabolism that might be responsible for poor outcome, but also maybe a different switch to another metabolism that might explain good outcome. And that was our hypothesis, actually, intrigued from all this data up to now with biomarkers, with metabolism, with maybe brain recovery, wanted to explore what if a switch to a different metabolic pathway may play a role in recovery, and not just look the poor outcome, but try to find who is gonna recover. So our hypothesis is that the switch to different metabolic pathway may play a role in outcome in non-traumatic acute brain injury. And how to look into that, initially, we want to start from humans and take CSF, collect CSF from patients suffering from non-traumatic brain injury, meaning subarachnoid, massive stroke, and ICH. So our aims would be to try to identify maybe a different metabolic pathway, a switch to a different metabolic pathway that would explain recovery and could be a predictor of recovery. So our primary outcome, of course, would be the 3 months outcome prediction. Secondary would be if we find a way to predict ICD treatment response and shunt dependency. Of course, it's not going to be an invasive study, meaning that we plan to take CSF from patients that they're going to have an EVD. So our inclusion criteria will be every adult patient come to the ICU, either at Mount Sinai West or Mount Sinai Hospital, that they have a non-acute brain injury, a non-traumatic brain injury. That's my mistake here, but non-traumatic, no. And then they're going to have an EVD. for whatever reason. If they have an EVD, we're going to include them. If they don't have an EVD, despite of their pathology, even if they have LP, we're not going to include them. We're going to include also some controls, patients that they come for lama drain, for high pressure, for hydrocephalus treatment, or for any other reason of treatment that they might have a lama drain, or EVD for another reason, like post-op sometimes, like we might want to include these patients for normal control. Of course, before doing anything, we're going to discuss with primary attending of patients, like primary surgery attending, to see if that's in agreement with the treating team, that they give us the green light to proceed and take consent from the patient or from the next of kin. Of course, because it's how it is designed, the study, the consent has to be in person, and IRB is monitoring very close that. Of course, we're going to exclude any minor patient, and TBI we decided to exclude because we don't have so many TBI. So even if we include TBI, it will be very like a lot of variability, and they have a different mechanism also of recovery and function. Patients also with primary secondary brain tumor will be excluded for obvious reason. Also, in order to avoid any increasing any risk, we're going to do the collection from CSF from the burette. So it's exactly kind of what the MGHs have done. So we're going to take CSF here from the needle side of the burette. So we're not going to flash, we're not going to touch any of the tubes or any of the connection between patient and AVD. Really, we're going to take from the burette just before the nurse open to empty the CSF that it's in the burette. We're just going to take it just before the nurse toss it out. So we're going to do it in collaboration with the nurse. We're going to know what time she empty, because I know they empty it every hour. So we're going to try to coordinate with the nurse what time she emptied. So we know she emptied at 10 a.m. Okay, we're going to go by 11, before 11, so we can collect the CSF at this point. And then we aim to do it in 3 different time periods, between day 0 and 2, between 4 and 6, and the day of EVD removal, or if the day of shunt placement, depends if the patient's going to have a shunt. For the moment, the samples immediately after be taken, they are going to be mixed, centrifuges, aliquoted, and stored at -80. We're going to store them. Dr. Kellner volunteered to give me some space in his freezer, because I don't have any freeze myself, of course. So they're going to be stored at Dr. Kellen's lab, and then we're going to analyze down the road. And mainly, my plan is mainly to do metabolomics analysis. Of course, we want also to check, to do multiplex ELISA technique, or maybe SOMA technique, to check inflammatory and non-inflammatory proteins. And of course, other techniques may be done down the road. So we're going to try to take enough CSF to have further for whatever technique we want to analyze. And the timeline of this project is going to approximately take at least 2 to 3 years, even longer, right? But to start with, it has been taking more than a year to even have under IRB review. So it's going to be a long process, but we anticipate the next months, pending IRB approval, we're going to start enrolling slowly patients, currently applying and looking for different fundings, and waiting for funding to have them. That's why this sample is going to be stored. Once funding is secured, then we can start doing some preliminary analysis, and then hopefully by second year, we're going to have some preliminary analysis and apply for further funding, and then continue collecting patients. And I guess by the end of third year, we're going to have a better idea and more more data, and we can come to a conclusion. Of course, down the road, goal to do that, because the population is going to be very heterogenic, and it will be only able to make association, not causation, right? So in theory, ideally, it'd be nice to, whatever data we have in the next years, to be able to implement an animal model, but that's going to be for years after. Big thing to discuss, right? We discussed clearly different surgeons, so we don't increase any infection risk or any other side effect other than the usual risk that are associated with NEVD care. We're going to be very careful to avoid any increased risk of infection, and that's why we're going to take the CSF from the burette, even though it's not the best CSF to take, because it might sit in the burette a little bit. But of course, our priority is no harm, so that's why we're going to take the CSF from the burette. It's going to be cleaned and taken by a needless syringe. Of course, it's in one way, so we don't induce any infection. Of course, we're going to take CSF only when the AVD is open and draining. If for whatever reason the AVD is clamped, we're not going to unclamp it to take AVD. So we're not going to increase any risk of overdraining or mess with the treatment of the patient. And of course, there's going to be no flush or anything, so we're not going to insert any air or do anything harm. So it's just going to be an aspiration without flush, without air insertion, and only from the burette, so we're not increasing the risk of infection. Neurosurgery Feasibility Committee has approved this project in early February, so currently it's under IRB review and funding seeking. And that's it. Thank you very much for your time, and I would really appreciate if you have any comments or questions.
Great presentation. Chris, are you part of this, or you want to comment or question?
Absolutely. One thing, Spyridoula, I think that would be really interesting is to see if you're able to develop a signature for neuroprotection that could be a surrogate outcome measure in studies. You know, as we start doing neuroprotection after ICH or after subarachnoid hemorrhage for vasospasm, to show some kind of surrogate marker or biomarker of that neuroprotection being effective. That would be, I think, really, really helpful.
Yes, thank you very much. Yeah, that's what we hopefully.
Well, that's a fantastic, interesting idea, but how can you show that if the protection treatment may last a few hours and your sampling frequency is at 0 to 2 days, on a daily basis? So how would you show the difference between before and after treatment then with that sampling frequency and the likely interventions that you would be doing, Chris?
I can imagine if you had a patient with cerebrospinal fluid continuously draining, and that patient got verapamil and they had some symptomatic vasospasm, went down to angio, got verapamil, and you had a pre and a post CSF from that, and you were able to see the patient got better, no longer had symptomatic DCI, and then you saw changes in the cerebrospinal fluid inflammatory
profile.
That's one example. I think trying to identify that and work through that problem would be really helpful to future trials.
Correct. And I mean, I put to collect CSF only these 3 time periods, also like to be practical and with IRB and be very vague, because there are going to be a lot of difficulties in the beginning. I'm the only one, right? So I'm not available 24/7 for every site. So that's why we said, like, with IRB, like, and Dr. Hadjipanagis, like, advice, like to do it very, like in open, like to leave it more vague, because I might be available day 0 and also can take more samples, but I may not for whatever reason. So that's why we leave it like that to start with. But of course, down the road, securing funding or even like people to help, then we may take more frequent samples. But it's like to start with, where we would say to do it like that.
I see. So the way that you've listed it will determine the minimum amount of samples you can take, but not the maximum.
Not the maximum, it's the minimum.
I see.
And then if we see something, I can do addendum with IRB and take more. But like it was to start with and to show that, because if I say that we're going to do more and at the end I cannot, then it's going to be bad, right? So it's at the minimum that I can assume that I can take and I can like guarantee that I can do, and then down the road we're going to increase. But that's the minimum amount of CSF we say to collect.
Is that what that means, Kostas, on the IRB application, that if you put that down, then that's the expectation of a minimum?
That's a good point. I'm going to emphasize that for the IRB.
Spyridoula, that sounds like a very, very cool study. 2 questions that I had. Dr. Connor kind of already brought one of them up, just in regards to the design. Certainly, I think the frequency, addressing that is going to be an issue, because if you're only looking at 2 time points with the tests that you specified, I mean, there's a high chance that you may just not catch what you're trying to catch, especially if you're looking at, markers of cerebral vasospasm development, for instance, right? And that develops over, 10 to 14 days, peak maybe, or over that duration. you might be missing something, and then your preliminary study, you might be on the right track, but you're just not going to capture it. And so your 10 patient preliminary study might be impacted by that. So maybe spacing it out if you can only do 2 time points other than just, so early. The other concern that I would have is about the design of drawing from the burette. I know you said that other people have done that, but it really is going to be a big factor in how long that CSF has been sitting in the burette. If you're trying to look for anything like protein or, especially something like RNA,
it's going to be
it's going to be just junk, and you probably, I wouldn't believe the results, and you have to really draw it very fresh. And so whether or not you run out more fluid into the burette to get there, you kind of have to maybe like just trying to get rid of some fluid to start.
That's a good point.
Or you take the risk and draw closer to the source. That would be a way to try and help that.
Yeah, that's a very good point. So that's why, I think it's really we don't want to increase any potential risk, because if I try to take from a point, even though it's always sterile, you increase a potential risk of infection or complication. That's why we said to start with burette. But I agree with say, if I don't take it, if the nurse emptied at 10 a.m. and I go at 10:55, then it's been sitting there for 55 minutes, so it's going to be junk. So it really has to be. I'm going to try to coordinate with a nurse, so the moment that she emptied, I can stay there and take fresh CSF going to the burette before sitting there for a whole hour. But yeah, that would be a big challenge.
I would also just keep in mind, Spyridoula, that everything dripping into the burette has already been in the tubing for a while, because it's progressive.
But would you recommend to take as we take CSF to send flash, that wouldn't increase the risk of?
I would actually, if you're able to design a protocol that allows you to clamp the EVD for half an hour and then drop it and quickly drain a little bit of CSF and then take it out of the burette, you could do something like that. So you're taking it further from the source, but it's fresher.
Okay.
Just
so would you recommend to clamp the EVD for half an hour and then
you just quickly drain some out and then take it from the burette? It's a little more labor intensive, but it's probably
if it's surgery, I guess depends on the patient profile.
I think we can
safe to do it.
We can probably talk about this offline.
Yeah, perfect.
Plenty of good ideas to.
I'm glad to hear. But thank you.
I think it would be okay if you emptied the burette and then closed it so that it was draining and collecting new fresh fluid, and then you had a certain amount that you want, like 3 cc's or 5 cc's. And then you take that fresh 5 cc's that have passively drained out. I think that's probably the least
safest
interruptive mechanism to give you.
So directly from the burette, once it's clear, don't just take the fresh CSF from the burette, you say. Okay.
That's what I would say. That's what I would suggest. But I think you've got to come in with a few ideas of what you want to isolate, and you're going to have to search for those things. You're going to have a lot of patients with a lot of data, but you're going to have to really hunt for 10 of a particular
that
you can compare.
I think we're going to have to move on. I think Dr. Batterson had a couple of slides.
Maybe
thank you very much, Dr. Tsetsou.
Maybe what you learned from the peri-clot analysis would be relevant.
Yeah.
To look at in CSF as well.
Okay, thank you. Yeah, hold that.
Great. Exciting work, and we look forward to hearing more. Really glad you're doing this. Okay. If you will